What is DNA? DNA stands for deoxyribonucleic acid-trusted source. It contains units of biological building blocks called nucleotides. DNA is a vitally important molecule for not only humans, but also most other organisms. DNA contains our hereditary material and our genes, the things that make us unique. What is the structure of DNA? A collection of nucleotides makes a DNA molecule. Each nucleotide contains three components: a sugar, a phosphate group, a nitrogen base. The sugar in DNA is called 2-deoxyribose. These sugar molecules alternate with the phosphate groups, making up the backbone of the DNA strand. Each sugar in a nucleotide has a nitrogen base attached to it. There are 4 different types of nitrogen bases in DNA. They include adenine, A, cytosine, C, guanine, G, thymine, T. The 2 strands of DNA form a 3D structure called a double helix. When illustrated, DNA looks like a spiral ladder in which the base pairs are the rungs, and the sugar-phosphate backbones are the legs. Additionally, it's worth noting that the DNA in the nucleus of eukaryotic cells is linear, meaning that the ends of each strand are free. In a prokaryotic cell, the DNA forms a circular structure. DNA explained and explored. Medically, what is DNA? Structure, function, DNA and health, DNA in cells. Takeaway: why is DNA so important? Put simply, DNA contains the instructions necessary for life. The code within our DNA provides directions on how to make proteins that are vital for our growth, development, and overall health. Keep reading to discover more about the structure of DNA, what it does, and why it's so important. What does DNA do? DNA contains the instructions that are necessary for an organism to grow, develop, and reproduce. These instructions exist within the sequence of nucleotide base pairs. DNA helps your body grow. Your cells read this code 3 bases at a time to generate proteins that are essential for growth and survival. The DNA sequence that houses the information to make a protein is called a gene. Each group of 3 bases corresponds to specific amino acids, which are the building blocks of proteins. For example, the base pairs TGG specify the amino acid tryptophan, while the base pairs GGC specify the amino acid glycine. Some combinations, like TA, TAG, and TGA, also indicate the end of a protein sequence. This tells the cell not to add more amino acids to the protein. Proteins contain different combinations of amino acids. When placed together in the correct order, each protein has a unique structure and function within your body. How do you get from the DNA code to a protein? First, the 2 DNA strands split apart. Then, special proteins within the nucleus read the base pairs on a DNA strand to create an intermediate messenger molecule. This process creates the messenger molecule RNA, mRNA. mRNA is another type of nucleic acid. It travels outside the nucleus, serving as a message to the cellular machinery that builds proteins. In the second step, specialized components of the cell read the mRNA's message 3 base pairs at a time and work to assemble a protein, amino acid by amino acid. This process is called translation. DNA in health, disease, and aging. The complete set of your DNA is called your genome. It contains roughly 3 billion bases trusted source, 20,000 genes, and 23 pairs of chromosomes. You inherit 1/2 of your DNA from your father and 1/2 from your mother. This DNA comes from the sperm and egg, respectively. Genes make up very little of your genome, only 1%. The other 99% helps regulate things like when, how, and in what quantity your body produces proteins. Scientists are still learning more and more about this non-coding DNA. DNA damage and mutations. The DNA code is prone to damage. According to estimates, tens of thousands of DNA damage events occur every day in each of our cells. Damage can occur due to errors in DNA replication, free radicals, and exposure to UV radiation. Your cells have specialized proteins that can detect and repair many cases of DNA damage. There are at least 5 trusted source major DNA repair pathways. Mutations are permanent changes in the DNA sequence. Changes in the DNA code can negatively impact how the body produces proteins. If the protein doesn't work properly, diseases can develop. Some diseases that occur due to mutations in a single gene include cystic fibrosis and sickle cell anemia. Mutations can also lead to trusted source the development of cancer. For example, if genes coding for proteins involved in cellular growth mutate, cells may grow and divide out of control. Some cancer-causing mutations are heritable, while others develop through exposure to carcinogens like UV radiation, chemicals, or cigarette smoke. But not all mutations are bad. Some are harmless, while others contribute to our diversity as a species. Changes that occur in at least or more than 1% trusted source of the population are called polymorphisms. Examples of some polymorphisms are hair and eye color. DNA and aging. Unrepaired DNA damage can accumulate as we age, helping to drive the aging process. Something that may play a large role in the DNA damage associated with aging is damage due to free radicals. However, this one mechanism of damage may not be sufficient to explain the aging process. Several factors may also be involved. One theory trusted source as to why DNA damage accumulates as we age concerns evolution. It's thought that DNA damage is repaired more faithfully when we're of reproductive age and having children. After we've passed our peak reproductive years, the repair process naturally declines. Another part of DNA that may be involved in aging is telomeres. Telomeres are stretches of repetitive DNA sequences at the ends of your chromosomes. They help protect DNA from damage, but they also shorten with each round of DNA replication. Studies associate telomere shortening with the aging process. Some lifestyle factors such as obesity, exposure to cigarette smoke, and psychological stress can also contribute trusted source to telomere shortening. Where is DNA found? DNA is present in our cells. The exact location of it depends on the type of cell. Eukaryotic cells. Humans and many other organisms have eukaryotic cells. This means that their cells have a membrane-bound nucleus and several other membrane-bound structures called organelles. In a eukaryotic cell, DNA is within the nucleus. A small amount of DNA is also in organelles called mitochondria, which are the cell's powerhouses. Because there's a limited amount of space within the nucleus, the body condenses the DNA into packages. There are several different stages of packaging. The final products are the structures that we call chromosomes. Prokaryotic cells. Organisms like bacteria are prokaryotic cells. These cells don't have a nucleus or organelles. In prokaryotic cells, DNA resides in the middle of the cell, called a nucleoid, coiled tightly. What happens when your cells divide? Your body's cells divide as a normal part of growth and development. Each new cell must have a complete copy of DNA when this happens. To achieve this, your DNA must undergo a process called replication. When this occurs, the two DNA strands split apart. Then, specialized cellular proteins use each strand as a template to make a new DNA strand. Following replication, there are two double-stranded DNA molecules. One set will go into each new cell when division is complete. Takeaway: DNA is pivotal to our growth, reproduction, and health. It contains the instructions necessary for your cells to produce proteins that affect many different processes and functions in your body. Because DNA is so important, damage or mutations can sometimes contribute to disease development. However, it's also important to remember that mutations can be beneficial and contribute to our diversity. What is autosomal DNA and what can yours tell you? Dominant and recessive. Examples: testing, takeaway. Nearly everyone, with rare exceptions, is born with 23 pairs of chromosomes that were passed down from parents through combinations of their 46 chromosomes. X and Y, the two most popularly known chromosomes, are part of the 23rd pair of chromosomes. They're also called the sex chromosomes because they determine what biological sex you're born with. However, this binary is not as simple as it seems. The rest of the 22 pairs are called autosomes. They're also known as autosomal chromosomes. Autosomes and sex chromosomes contain a total of about 20,000 genes. These genes are essentially 99.9% trusted source identical in every human being. But small variations in these genes determine the rest of your genetic makeup and whether you inherit certain traits and conditions. Autosomal dominant versus autosomal recessive. Within these 22 autosomes are two categories of genes that pass on different traits and conditions from your parents. These categories are called autosomal dominant and autosomal recessive. Here's a quick breakdown of the difference. Autosomal dominant. With this category, you only need one of these genes to be passed on to you from either parent to receive that trait. This is true even if another gene in the same autosome is a completely different trait or a mutation. Inheritance. Let's say your father has just one copy of a mutated gene for an autosomal dominant condition. Your mother doesn't. There are two possibilities for inheritance in this scenario, each with a 50% chance of occurrence. You inherit the affected gene from your father as well as one of your mother's unaffected genes. You have the condition. You inherit the unaffected gene from your father as well as one of your mother's unaffected genes. You don't have the condition, and you aren't a carrier. In other words, you only need one of your parents to pass an autosomal dominant condition onto you. In the scenario above, you have a 50% chance of inheriting the condition. But if that one parent has two affected genes, there's a 100% chance you'll be born with it. However, you can also get an autosomal dominant condition without either parent having an affected gene. This happens when a new mutation occurs. Autosomal recessive. For autosomal recessive genes, you need one copy of the same gene from each parent for the trait or condition to be expressed in your genes. If just one parent passes on a gene for a recessive trait, such as red hair, or condition, such as cystic fibrosis, you're considered a carrier. This means that you don't have the trait or condition, but you may have the gene for a trait and can pass it on to your children. Inheritance. In the case of an autosomal recessive condition, you need to inherit an affected gene from each parent in order to have the condition. There's no guarantee that will happen. Let's say both of your parents have one copy of the gene that causes cystic fibrosis. There are 4 possibilities for inheritance, each with a 25% chance of occurrence. You inherit an affected gene from your father and an unaffected gene from your mother. You are a carrier, but you don't have the condition. You inherit an affected gene from your mother and an unaffected gene from your father. You are a carrier, but you don't have the condition. You inherit an unaffected gene from both parents. You don't have the condition, and you aren't a carrier. You inherit an affected gene from both parents. You have the condition. In this scenario where each parent has one affected gene, their child has a 50% chance of being a carrier, a 25% chance of not having the condition or being a carrier, and a 25% chance of having the condition. Examples of common conditions. Here are some examples of common conditions in each category. Autosomal dominant. Huntington's disease. Marfan syndrome, blue-yellow color blindness, polycystic kidney disease, autosomal recessive, cystic fibrosis, sickle cell anemia, Tay-Sachs disease. About 1 in 30 Ashkenazi Jewish people carry the gene, homocystinuria, Gaucher's disease, autosomal DNA testing. Autosomal DNA testing is done by providing a sample of your DNA from a cheek swab, spit, or blood to a DNA testing facility. The facility then analyzes your DNA sequence and matches your DNA to others who have submitted their DNA for testing. The larger a testing facility's database of DNA, the more accurate the results. This is because the facility has a larger pool of DNA for comparison. Autosomal DNA tests can tell you a lot about your ancestry and your chances of getting certain conditions with a pretty high level of accuracy. This is done by finding specific variations in your genes and putting them in groups with other DNA samples that have similar variations. Those who share the same ancestors will have similar autosomal gene sequences. This means that these DNA tests can help trace your DNA and the DNA of those distantly related to you back to where those genes first came from, sometimes back several generations. This is how these DNA tests can suggest your ancestral ethnic and racial background, trusted source, and what regions of the world your DNA comes from. This is one of the most popular uses for autosomal DNA kits from companies like 23andMe, AncestryDNA, and MyHeritageDNA. These tests can also tell you with almost 100% accuracy whether you're a carrier of an inherited condition or have the condition yourself. By looking at the traits within the genes on each of your autosomal chromosomes, the test can identify mutations, either dominant or recessive, associated with these conditions. The results of autosomal DNA tests can also be used in research studies. With large databases of autosomal DNA, researchers can better understand the processes behind genetic mutations and gene expressions. This can improve treatments for genetic disorders and even lead researchers closer to finding cures.